All right, we're going to get started here. Hello, and thank you guys for joining us in Track 4 today in the Science and Technology Innovation, sponsored by PRA Health Sciences. My name is Jessica Shiles, and I will be your room host today. And just as a reminder, this track is being live streamed, so I just want to welcome all of our remote attendees. And I would also like to remind everyone to go ahead and download the mobile app. You can go ahead and see some of our features and highlighted features, as well as be involved in the live question and answer. So today's breakout session will be presented by Bartholomew Tortella, the Global Medical Affairs Lead of Rare Disease Hematology and Transplantation at Pfizer, and Alex Mori, Medical Science Liaison at Alnylam Pharmaceuticals. Today, we are actually going to be going over the promise of 2 different rare disease gene therapies that look at adding either the correct copy of a gene or altering mutated copies of a gene during this session, which is titled Rare Gene Therapies, A New Era for Genetic Medicine. So please welcome me in joining both of our speakers.
Thank you, Jessica, and thank you all for being here. As Jessica said, Bartholomew Tortella from Pfizer, Rare Disease, a division within Pfizer. And then there's a disclosure. Everything I say is my opinion, does not necessarily reflect Pfizer's. And I was talking with Alex before we began. Gene therapy is not something in the future. The first gene therapy in the United States was approved a few months ago to treat blindness from a specific genetic mutation. And Pfizer itself is embarked on right now a phase 3 gene therapy trial in hemophilia B. So I think gene therapy is particularly important because it's not something that is coming, but rather that is here for blindness and for a number of other genetic diseases are all now in pivotal trials. So it's particularly important because gene therapy is going to be a tool and is now a tool in the toolkit for physicians and patients that they can use going forward. So I'm going to organize around the principles of adult learning. So I'm going to tell you what I'm going to tell you. I'm going to tell you and tell you what I told you. So first, starting with proteins, very important. They're building blocks of life. Second, what about those vectors? What are they like and what do they do? And then finally, the catch. Gene therapy is very simple to talk about, but Dr. Miller in his previous talk started to talk about some of the issues that gene therapy has with the immune system. So everyone knows about proteins. You learned about them in basic biology, and they're pieces of amino acids put together, and they hold us together. They change us, and they allow us to live. And everyone here knows that one of them, if they're absent or malfunctioning, or if there's too much of them rather than too little, can cause disease. So gene therapy really right now is about focusing on proteins and how to provide them. So where do they come from? Well, I think everybody remembers that proteins come from your genes, and the genes have an alphabet, and it spells out words, and those words gets turned into protein. And if the words are misspelled or the words are absent, then we can't make those proteins. And there's some new words. The slides will be available, so I'm not going to cover every individual thing. But some words you need to be aware of is the transcription, which is taking the gene and making it into a protein. We're going to each take a look at those segments and see how gene therapy affects them. The first thing is, where is the working part of gene therapy? How does that look? And it's really composed of 2 things. First of all, the transgene. The gene we're transferring, gave it a fancy name, transgene. In my case, hemophilia, it makes a specific protein, as Dr. Miller said, which allows the blood to clot. But the gene itself is not enough. You can see there in white the promoter. The promoter is a special set of instructions which tells the protein-making machinery to to pay attention to the gene right behind me. Make a lot of the protein from the gene behind me. And the nice thing about these promoters is they are specific to different organs in the body. So for hemophilia, these proteins are made in the liver. So the promoter that we select tells the liver, make a lot of the protein using the gene right behind me. For Duchenne's, Pfizer has a gene therapy trial in Duchenne's. It's a different promoter. It's a promoter for muscle. So we can control how much of the protein is made based upon the type of promoter we use, and that can be for the liver, or be for the muscles, or be for the brain. So the gene inside the gene therapy is not just the instructions to make the protein. There's also that promoter which allows us to control how much of the protein is made and exactly where it is made. So that's the gene. How do we get it into the cells? Not into the cells, but into the right cells. Okay. And here we are borrowing from nature. Viruses in nature bring their DNA into the cell, cause the viruses to produce, and then new viruses are released from the cell. So this is nature's own way of transferring DNA into the cell. So here's a new word for you. Capsid. Not a capsule, but a capsid. So if you can think of a soccer ball made of protein, we manufacture the protein in our factory, and that protein ball is the capsid. Inside, we place the gene and the promoter that we just talked about. We put the capsid together with the transgene, and we get the vector. So we talked a little bit about the transgene. Let's now talk about that vector. First of all, we are not infecting patients with gene therapy. That requires a virus. We do not use a virus, we use a capsid. It is drawn on the blueprints of a virus, but itself is not a virus. This is what a virus does. It goes in, multiplies, and then is released. Gene therapy is a one-way activity. The vector goes into the cell, releases its gene, and the gene does the work there. So we don't infect patients, we treat them with our gene therapy. And this is a description of what it looks like. The virus goes inside the cell, the virus opens up, the transgene comes out, and that transgene is delivered to the liver cell, the muscle cell, or the nerve cell. You remember capsid, right? That's that soccer ball. This process is called transduction because we are giving the cells a piece of DNA that they can use to make the protein. Now, there are many different ways that you can get the DNA into the cell, and as Dr. Miller said, there is a way to insert the gene into the DNA, but you have to be very, very careful. If you insert it at the wrong place, you could damage the cell and damage the genes. So many gene therapies, Pfizer's, the one for the eye, are simply adding an extra piece of DNA into the nucleus. As Dr. Miller said, sometimes when that cell divides, the chromosomes in the nucleus divide, but these extra little pieces of DNA that we provide don't always divide. And we have a name for that extra little piece, and that's an episome. So in gene therapy, we take the transgene inside the capsid, called a vector. The vector goes into the cell, the soccer ball opens up, the transgene goes out and into the nucleus and stays there as an episome. Many different platforms that we can use. The one that is most commonly used for adding that piece of DNA is adeno-associated virus. Not the cold virus, that's adenovirus. Adeno-associated virus does not cause any disease. You don't get a headache, you don't get fever, you don't get chills. It's all around us. It's, in fact, the older you are, the more likely it is you've been exposed. So the lentivirus are those type of treatments which occur outside the body, as Dr. Miller said. We're going to look more closely at the adenovirus, adeno-associated virus. And the reason we like that one and is being used and has been approved by the FDA for the blindness is that it is predominantly non-integrating. It doesn't put that DNA into your chromosomes. It leaves it separate. And that way we minimize the chance of landing somewhere in the DNA that we didn't intend. So a separate piece called an episome. Sounds very straightforward, but as they say in the business, there's always a catch. And the catch is the immune system. That always stands guard and really inspects all the medicines that we give, however we take them. And it has two components that are important for gene therapy. The first is the rapid response, and the second are the reinforcements. Dr. Miller spoke a little bit about the rapid response. Those are your antibodies. And the antibodies are circulating in your blood right now. And if you get something that exposes the antibody and it binds, the antibody blocks it. Anybody who have an allergy? You have antibodies circulating in your system. When that item that you're allergic to comes, the antibodies bind to it, and then you get the allergic reaction. You don't get the allergic reaction with gene therapy, but if you've been exposed to AAV, and our vector looks a little bit like that AAV, the antibodies block the gene therapy from getting into your cells. So actually the first thing we do if a patient comes and wants to enroll in a clinical trial is we test their blood to see if they have these blocking antibodies. If they do not, then they are eligible to get into the trial, and if everything works out, the product would be approved. This would be the first thing that a patient and a physician would do together if the patient was interested in gene therapy. This also means that if you're without those antibodies and you can take the therapy, right now you only get one opportunity. Because if you did not have the blocking antibodies before, and we give you the therapy, your immune system makes the blocking antibodies. So where is research in gene therapy right now? Mostly immunology. Here's the way we can decrease the number of antibodies so that we can treat patients who have the antibodies. Or is there a way we can prevent the body from producing so that we can treat 2 or 3 different times with gene therapy? That's where the research is. Right now, it's a single treatment. And the second thing is those T-cell responses. So here's a picture of how the antibody would bind the virus and prevent it from getting into the cell, and even low levels will prevent the gene therapy from being effective. So it's important to have a very good test to see if your body has those blocking antibodies. And the second thing is those long-term effects of the T cells. The T cells are constantly surveilling the body for unusual or infected cells. Well, we know our gene therapy cells aren't infected, they're transduced, and they're making the protein, so it's actually a good thing. But the immune system doesn't know that. So sometimes if it finds one of these cells that it is now making the protein, it can attack and destroy that cell. So we watch very carefully for the first 12 weeks when the immune system is on high alert. If we see one of these attacks taking place, we give oral prednisone or steroids for a number of weeks. That quiets the immune system and allows the cells to continue making the protein without further damage. This is a little picture of what we think it looks like, where the vector goes in, pieces of that soccer ball are cut up, put on the surface of the cell, where the T cells see them and start their attack. And if we give prednisone, we can stop that attack and preserve those cells which have now taken in the gene and are producing the protein. We take the in vivo approach, as Dr. Miller said. It's a simple injection, takes about an hour. Patients go home the same day. The outside the body, more complicated, and he talked about the need for chemotherapy. We have focused on the in vivo or injection of the gene therapy. So that's a quick walk through gene therapy from the standpoint of what actually is being done now for congenital blindness and what many companies are doing for gene therapy for genetic diseases. I'd like to thank you for your attention and hand it over to Alex.
Thank you. So, I don't have principles of adult learning in my slide, but I'm going to take advantage of everything Bartholomew just did, because I think that was a wonderful primer. And actually, I mean, my story is a little bit of a different story. I'm going to tell this more sort of along the lines of a journey that the company that I work for has been on for a number of years. But to put the story into context, to understand what Alnylam and all of the companies in the genetics space are trying to do, I think it's important that we understand just how extraordinary it is that we're on the cusp, we're beyond the cusp, of a different way of looking at medicine. And the truth is that if we look across medicine, right, there aren't many new things under the sun. Vaccines, right, before the 1700s, right, the only thing you had was leeches and surgery. That's it, and that's the reason we all died in our 30s. And all of a sudden somebody discovered that there's a way in which I can pre-expose you to something and we can vaccinate and prevent disease before it begins. Now, how many of us have had vaccinations, right? We've all had vaccinations. We all know people who had vaccinations. So this is commonplace now in medicine, but it wasn't in the 1700s. And the reason that I'm showing you this is because I want you to see how long it takes between new innovative breakthroughs. It took 100 years before we started thinking about small molecules. Tylenol, Advil, we take them, we don't even think about it. But yet oral small molecules change the way we treat medicine, disease. 40 years later, antibiotics come on the scene. 50 years later, monoclonal antibodies. But take the monoclonal antibody as an example. So mouse antibodies were discovered in the mid-70s, 1975. The person who discovered the mouse antibody and described its function, this protein, won the Nobel Prize 9 years later. But it still took till 1998 before we actually had a therapy that actually could take advantage of those proteins. So over 20 years of R&D and development. And so when you take a look, fast forward 40 years, right? So here we are at present, and we talk about these gene therapies, and we have companies that are doing gene therapy and gene editing, cellular therapies, antisense therapies, CAR T therapy, all this stuff, right? I want to give you a sense of how rare it is that we have a new paradigm to talk about medicine. And I also want to give you a sense of the fact that there's a lot of research that has to go in. I mean, and as we saw with antibodies, 20, 30 years of research to actually realize the potential of the technology. So I've heard from patients in the past, didn't we discover what the human genome had, a decade ago? And it's true, we did. But there's a lot of smart people who are trying to make this a reality. And the company that I work for is really focused and has been focused for the last 16 years on a very small subset of gene therapy. We're a company that's interested in a process called RNA interference. And in order to understand how that process works, I have to take you back a couple of years to 1990 and an experiment that was done at the University of Arizona by a bunch of botanists that were interested in petunias. Now, in 1990, not much was known about genetics, but they did know that the purple petunia created a gene that gave it its purple color. So they wanted to ask a very simple question. They said, look, if I take purple, the gene, and I put it into a purple petunia, what should I get? Right? Probably a more purple flower. That was sort of the idea, right? One copy of purple is purple, 2 copies of purple should be more purple. Well, it turns out what they got was a white petunia. Now, here's the thing. This doesn't make any sense. So the only thing they could say was there must be some sort of mechanism to suppress gene expression that we don't really know about. And like with a lot of great discoveries, it sat on the shelf for 8 years, right? This was known in botany. It was an interesting curio, but people really didn't know what to make of this. And they started looking at it. And in 1998, they actually proved that this can work. The same process that's working in flowers also works in worms. It doesn't sound like that big of a deal. Who cares about a worm? The reason it's important is because a worm looks a lot more like a human than a flower does. And then in 2001, they proved that this could actually work in humans. So it is possible to introduce a genetic element into a cell and actually suppress gene expression. And so when we think about paradigms and we think about really changing the way we think of medicine, right? A lot of the therapies, until the advent of personalized medicine, really can only treat symptoms, right? And that's like saying, I have a dripping faucet, and I'm going to take a mop and bucket and wipe up the puddle. That's about as good as we can do. The problem, though, isn't with the puddle. The problem is with the faucet. And so what we're hoping to do with RNA interference technology is say, look, let's go to the root cause of the disease. Let's go to the faucet and shut that off. And if we do that, in theory, right, we should shut off the puddle too. So RNA interference and all the mechanisms through which RNA interference worked were discovered over a decade. And in fact, actually, the discovery of RNA interference won the Nobel Prize, and the description of the cascade won the Nobel Prize in 2006 for a pair of scientists called Fire and Mallow. And after we showed that this could work in humans in 2001, Alnylam was founded in 2002 with the idea of taking this innovative technology and bringing it into the clinic. I don't know about how many of you guys have heard of the guy who invented the light bulb, right? Edison. He said, I didn't invent the light bulb. He said, I invented 999 ways not to make a light bulb. Well, it turns out we have this wonderful idea. We have this company. We're going to do this big thing, right? And literally, it was 10 people in a room, and they said, we're going to bring this technology. We're going to put it in the clinic. And then they started trying to put it in the clinic. And for 15 years, they didn't make light bulbs. They made everything but not light bulbs. But they made progress. And I think that that's something that you have to realize, that it does take time, but research is incremental. And every time you say no, then that's a path that you realize is wrong and we have to try something else. And companies started this, big companies started this, and said we can't do it anymore. After 4 or 5 years, in 2006, a lot of large companies said, forget it. And we said, we're crazy, or maybe we're not, and we're going to keep going with this. But actually, it turns out that we're able to do this, and I'm going to show you some examples I'll show you at least one drug that we just recently got approved that proves that we can do this, and I'll show you a couple of other trials that we have that we're looking at developing this further with. But here's the thing. What we want, the promise for our medications is this. We want to be able to deliver a medication that gives you the ability of knocking down a gene of interest at a level that we choose to knock it down at. Everything from a little bit of knockdown all the way down to potentially complete suppression, over 99%. And that's going to very much depend on the type of disease we're talking about, right? We have to make sure that these medications clamp the disease down. So what I mean by that is, if you're producing a toxic protein, it doesn't help that we have that up and down pattern as you see in the upper right-hand corner. That wouldn't help, because then half the time you'd have the toxic. So this has to be a permanent knockdown. We want to make the drugs durable. So not an injection every day, but actually, in fact, some of our medications now can be dosed as infrequently as every 6 months. And we have to make this as simple as to administer as possible. Preferred subcutaneous, simple injection. So that's the promise, that's the hope. And I'm really pleased to be sitting here today to talk about one disease that I've spent the last 4 years of my life working on, hereditary TTR amyloidosis. This is a rare condition. It affects about 50,000 patients worldwide, and it's created because of a protein in the liver that can't stick together properly. The protein falls apart and begins to deposit around the body. This would be a condition, as I mentioned, that if we could knock down TTR, perhaps we could affect the outcomes. And in fact, actually, our first approved drug shows that we are effectively able to knock this down by about 85%, and over time affect polyneuropathy in these patients. Now, this is one disease. Another disease is acute hepatic porphyrias. So these are a type of ultra-rare orphan disease. They affect about 5,000 patients in the United States with sporadic attacks. And these patients essentially get pain, debilitating pain, it being described like fire in their belly. And oftentimes these patients go undiagnosed, they don't know what they have, they go into emergency rooms and get diagnosed as being drug seeking, or maybe, what would you say if somebody said, this is all in your head? Well, that's horrible, but we can actually test for this condition. It is genetic, and it's created by an overexpression of, or an inability to synthesize heme, and we can actually reduce a protein and potentially alter the course of this disease. And then I think something near and dear to my heart, I just actually recently had 2 kids. So I have an 18-month-old and a 3-month-old. And this is a disease called primary hyperoxaluria. And it does affect primarily a pediatric population, very rare, maybe 5,000 people across the United States and in Europe. But this rare autosomal condition actually leads to an inability to metabolize a protein called oxalate that begins to deposit in the children's kidneys and eventually destroys those kidneys. And then once the kidneys can no longer filter the protein, it starts to deposit all over the children's bodies and eventually will kill them. And so, again, another condition where if we could just knock down the production of oxalate, right, maybe we could alter the disease course. And the reason I bring these 3 drugs, or these 3 concepts to your attention is because we're actually in the process of doing phase 3 clinical trials in all 3 of these conditions. So, So where do we hope to go with this? Well, we hope to eventually get products on market, and I'm really pleased to announce that as of August of this year, we've gotten our first approved RNAi therapeutic, the first one that's ever been approved in the entire world, called Onpattro, to market. This has taken the efforts of physicians across the world, but most importantly, patients. 225 brave patients risked their lives to join our studies, and so I'm very pleased that they were courageous enough to do this, and we continue to do this type of research because of the types of patients that are willing to participate. And so I just want to end here, just showing you some of our pipeline, just to give you a sense of the type of conditions that we are looking to treat with this type of technology. Obviously, I already mentioned the first couple, hereditary TTR amyloidosis and acute porphyrias. We are looking at rare bleeding disorders, like hemophilia. We are looking at hyperoxaluria and complement-mediated disorders. But again, this technology isn't just limited to rare diseases. A lot of us have hypercholesterolemia, and so familial hypercholesterolemia affects hundreds of thousands of people across the world. And for example, inclisiran is a way to knock down PCSK9. And so again, we have antibodies that can do that, but what about this technology? What if we could apply this technology? So I think the application of the technology is broad, and I'm really excited to be here today to talk to you guys and to share some of this information with you. Thank you.
So we're going to go ahead and open it up to question and answers. Does anybody have any questions for either Bartholomew or Alex? I can run the mic to you.
Thank you. So Alex, I know you'd mentioned with the RNAi knocking down or eliminating some of those proteins. Have you been able to find a way to increase the proteins? Let's say there's a gene that's not completely producing the right amount of proteins, only one of them's functioning, it's at half level. Mm-hmm. Is there a way to increase that with any of the pharmaceuticals or with the methods?
Well, RNA interference, for the technology that my company is working on, is an interfering technology. It knocks things down. But that's not to suggest we can't potentially think of creative ways to knock things up. So for example, in hemophilia, we know that patients don't produce enough clotting factor. So we actually target the antithrombin. So thrombin is usually produced in healthy people, helps you clot. But by destroying the destroyer of thrombin, you actually increase the thrombin production in these patients. And that might sound scary, but if you have a patient who produces almost no thrombin, we can, again, try to dial in the amount of suppression that we get for antithrombin and raise them, let's say, to the lower limit of normal. That would be the hope, anyways. So if we understand the genetic mechanisms through which the pathway works, and there's an inhibitor we can target, and it's sort of like the enemy of my enemy is my friend, right? The inhibitor of the inhibitor might increase gene expression. That's sort of an indirect way we could use this technology to increase gene expression. It's all on a case-by-case basis, of course.
Okay, any other questions? Nope.
Hi, I'm Pat Gibson. I work with the amyloidosis support groups. I'm their public policy liaison. And I just want to say to Alnylam, which has just given us the first treatment, which will help us to conquer our disease, that working with Alnylam, which is a very small, or has been, now it's bigger, drug company, we've been working with them since they started, that it has been a fantastic experience for us to work with a drug company who is willing to go to the patients, to work with the patients, to bring us in on their decision-making processes, and to have such a group of committed people from beginning to end. And I say to other patient groups, sometimes working with these small drug companies is really very valuable for you all. So thank you, Alnylam.
Thank you.
Hi, thank you very much for this talk. I'm Meryl Fink with the Progeria Research Foundation and have no medical background, so this may be a very simplistic question, but I hope others in the room may be thinking the same. I'm trying to be a little fearless here and be vulnerable and get out there. We've heard a lot about different types of what I group in my mind as genetic therapies. You talked about the RNA therapies, we've heard about gene editing. How do we know, or why would one work and one wouldn't? How would we know which one of these therapies to target?
That's a difficult question because each disease has its own personality, if you will, and its own speed of development. Hemophilia is present from birth and that protein is absent, but you've seen here many diseases you have to treat early in order to get the effect. And I think that's where the medical research comes in. You heard yesterday about many of the different research projects which are going forward. The nice thing about what you've heard today is we are now giving tools to the research community so that they can see, given this disease's tempo or the way this disease affects the body, which of these tools, is it the interfering RNA? Is it the gene addition? Is it the gene editing that needs to be applied and has the best chance of making a difference? So I can't answer a question in specific, but what I can say is we are providing the tools that the patients and the physicians and the researchers can use to see, taking a piece or a thought from each of these different approaches, what might be the best for that individual disease. The more tools, the more options, the more way to attack the program and the disease.
And I also wanted to comment on the importance, I think, of natural history studies. I think it's frustrating when you have a group of researchers that say, look, we want to study you and see how things go. But that's not because we don't want to help. It's because we don't know what we don't know. And sometimes with some of these diseases, there can be so few people who have ever had the disease that the whole medical community doesn't know. So natural history studies are a way in which we have to start there. We have to start with something so that we know if the therapies are effective. And so we understand the pathways through which the disease can be targeted. And then we can figure out what tools that we can apply from a chemistry standpoint to best target those.
Okay. Do we have any other questions? Oh. To you. Who was it that raised their hand?
Your steps in today. Hi, so I have a follow-up to the question in the front, and it might be very simplistic, excuse my ignorance. So if someone has developed an antibody or you've identified an antibody to the vector, is there a possibility that they could qualify for other means of gene therapy that might have been developed by another company?
That is an excellent question that we are now seeking to learn the answer. Because these gene therapies are so new, We don't know the amount of crossover or cross-reactivity. If you take a look at AAV, they all belong to the same family, but some are older adeno-associated viruses, some are newer, and I'm talking hundreds of millions of years as far as age is concerned. So it is possible, theoretically, that you might have antibodies to one family of the AAV, and we number them 1 to 9, and not have a neutralizing or blocking antibody to another. So it's probably going to be the case as these new gene therapies become available that you would be tested for the first one, and if you had the blocking antibodies, then you would wait for the second or the third, hoping that your specific antibody did not cross-react and that you would be able to take a different type of gene therapy. But because we are so early, it's difficult to say. There may be enough overlap so that if you have a blocking antibody to one gene therapy, you also may cross-react and that antibody may block the other gene therapy. Only time and medical science will tell us that answer.
Thank you. As the mother, fifth generation mother in my family due to narrative studies, to have children that are affected by genetic problems. I'm so thrilled with where you are, and I'm so thrilled that you're able to, both of you, explain this very complicated and miraculous new place we are in terms of medicine to all of us who are non-scientists. And I want you to answer a question which may or may not be appropriate here. So given, I understand what it cost in order to get where you are with that treatment and the size of the population that uses it. What have you guys had to do in order to make sure that the people who need the meds have access to it? Because that, of course, is my great fear. Mm-hmm. They're going to find a gene therapy cure. Mm-hmm. We know they are, to sickle cell disease, which is what I've passed on to my children. My great fear is they're going to find it, and then we're not going to have access to it. And so I'd like to know what your company is doing, because I know it's a small company, it costs a lot of money. I'd like to know what you're doing, and I'd like for you to tell the audience what we as advocates can do to help companies like yours and research going on, so that when you do the right thing and you get there, we're able to get it, and we've helped you with that.
So I think this is a very relevant question, especially in this day and age, right? We always hear about the increasing cost of healthcare. And I will say, from a company standpoint, I think that there's two things that I would advocate for the patients to do. If possible, make sure that you continue to do the advocacy work that you do, because payers, physicians who do this for a living, they don't know the diseases, right? And I can educate a physician. But a payer is, and it's not because of ignorance, right? They have 1,000 diseases that they're dealing with. So I think the more awareness we can bring around the disease, and the ramifications, what does this disease mean to a patient? That helps payers understand the burden of the disease. So here's an example. I helped support a drug for hereditary angioedema, which is a rare swelling disorder. You literally will start to swell and you can't stop. And sometimes it's in your foot and it's inconvenient. And other times it's in your foot and you can't walk for 3 days. And sometimes it's in your throat and you die. And this still happens. And I remember sitting in front of a payer, and I'm not going to mention names, but the payer didn't understand that, right? And he said, why would I pay for a little bit of swelling? We all have a little bit of swelling. And then I showed him pictures of patients. I said, this isn't a little bit of swelling. This is uncontrolled swelling 3 times a week in some cases. And he apologized. He said, I'm sorry, I didn't realize. So I think advocacy and making awareness is key. The other thing I think we can do better as companies is engage with the payers sooner. Nobody likes surprises, and making sure that they are aware and on board with what we're trying to do. I know that at Alnylam, we've been very proactive in creating what they call value-based agreements so that we can help offset some of the risk benefit, right, to the companies, and saying, look, we believe that we produce value, and we believe that that value should be, that's part of the American dream, right? We produce value, and that should be rewarded. But if we don't do what we say we're going to do, is there some way in which we can moderate the cost of those medications to help? And so far, we've seen a lot of payers be very interested in working with us to try to see if there are value-based agreements. But that all comes back down to what value does the patient get? And so if we think you really want this and you're going to really benefit from that, and we're sitting in cubes, you're going to get something that may or may not be valuable. But if we understand what the patient needs, wants, and what will really be effective in real life, that helps us structure those value-based agreements to help you.
And then from the gene therapy side, we're going to be a big problem for the healthcare system. Because the healthcare system is used to taking medicines that you take regularly, and then it finds a way to finance them. In gene therapy, we're talking about a one-time treatment whose durability may be years, decades, we don't know. How do you value 5 to 10 years of treatment all rolled in to one hour injection? So the healthcare financing system needs to adjust to a new wave of therapy, gene therapies, which are coming forward. The way they do that is by talking to the people in this room, and I'm looking to my colleagues from the National Hemophilia Foundation, again, my area is hemophilia, and they have been very proactive, starting with all their state and federal advocacy colleagues, and then their insurance advocacy colleagues. Because as Alex said, you have to be the voice in the room, because pharma is going to make its case, and then the healthcare finance is going to make its case. But the real case is the case made by the patients, because they are the experts in their disease. We only watch. We only attempt to help. The patient is the expert. They live their disease. So this is a great call to action to the advocacy community, that you must be in the room with the producers, with the payers, with the policymakers as an equal. I actually think you're more equal because you're the most important person, but use your advocacy voice to bring a realization of what the disease is, what benefit this treatment represents for you as a patient community. None of us here on the manufacturing side, no one on the policy side can do that. That's your unique voice, that's your unique responsibility, so that the medicines that are available are appropriately compensated and made available. So that's a great call to action to the group here. This is something only you can do.
Okay, do we have any other questions? I think we might have a few from the live question and answer. We're getting now. All right. So from some of our live streamers, it seems everyone is dreaming that gene therapy will cure our kids, but how can we as parents ensure that we have access to gene therapy when the number of diagnosed cases is low? More cases equals more money equals those diseases are treated first.
So first of all, I want to say we're not curing the disease in hemophilia. We're providing a durable treatment. But the genetic defect is still in the patient. When they have children, they can still pass on that defect. So we're a durable treatment. And I think so hemophilia is a rare disease, but that means there's 15 to 20,000, which is a huge number compared to some of the diseases in the room here. For the ultra-rare diseases, I think there is great hope. And the reason for that is a lot of the diseases we talk about, the congenital blindness, the hemophilias, the cystic fibrosis, where there's a larger number, are paving the way and perfecting the technology. So as you asked, what about for these rare diseases? Which one of these approaches is going to be? Well, these approaches are just getting started now. A few are approved and we're working our way through them. But for the ultra rares, I think you're going to benefit by the fact that the interfering RNA approach will be perfected. The gene therapy approach will be perfected. And so the ultra rare diseases will have a well-known way to incorporate the technologies about which we're talking today. And it's going to be difficult with such small numbers, with the development costs, but those costs come down as we gain more experience. Alex used the idea of monoclonal antibodies, which for 20 years ago were quite exotic and very expensive. Now we take monoclonal antibodies almost for granted. We can make them much more easily because over the last 20 years we've perfected that technology. So for the ultra rares, the real small diseases, the outlook is bright because the tools we're talking about here as they grow and are perfected will make it easier to access and apply to those ultra rare, but nonetheless very important diseases.
Thank you. Our next question is gene therapy lifelong or does the body begin to act normally?
We do not know. In hemophilia, the first patient ever treated was in the late 90s, and they are still making very low amounts of protein. The original publication for gene therapy was in 2010, 8 years ago, and those patients are still making low amounts of protein. So time will only tell. If you want to know about dogs and mice, they're treated for life when you give them the gene therapy, but we're not dogs or mice. We're not even non-human primates. We're something completely different. So we would look for duration of years to decades, but we do not yet know.
I have another question over here. I'll get to the next live question that was asked after this.
Hi, thank you so much for such clear, down-to-earth descriptions of such complicated science. I'm with the Jane Foundation. Talking about expression, are you working on any projects to quantify how much expression is meaningful for different diseases? We come up with this, we're measuring expression for dysferlin, which is a gene associated with limb-girdle muscular dystrophy type 2B. Adult onset muscular dystrophy, but we're wondering how much is going to be meaningful and is that expression even functional protein?
I think that's an important question that is being answered as we go forward. What Alex said is you need a registry, and then you need natural history, and then you need clinically meaningful endpoints. And the beautiful thing about the rare disease community is you take that on as a cause. So you'll fund individual laboratories which will try to establish what those clinically meaningful levels are. Alex said his technology can allow you to say, well, if you have 100%, we can drop you to 80, we can drop you to 60, we can drop you to 2. So we are now giving you the fine tuning necessary. The people in this room are those who are driving the research to answer those type of questions, because sometimes you may need to get to this low, or sometimes you may need to cut it off completely. But that's the unique beauty of this community. You seek out researchers and deliberately fund them with that very focused question. Because when it comes to larger pharma, the things we're going to want to know is, where are the patients, what do we need to do, and what is important to them. You've heard that the FDA now has patient centricity. And many of the endpoints for Duchenne's don't make sense when you ask the patients. The class is a 4-step walk test. And if you talk to Duchenne's patients, they really don't care about walking. They have an accommodation to walk. But can they feed themselves? Can they do daily activities with their arms? So one of the important endpoints for Duchenne's is not the medical endpoint of the 4-step walk, but some new type of endpoint that we can develop using the arms, because that's clearly one of the things we've heard from that community. So I would continue to do what you do and fund the research that asks those questions for Duchenne's or any of the other diseases, because that means when you come to us, you're fully enabled for us to say, okay, how many patients are they? Where are they? What do we need to do? What's been the research? Okay, we can take it and partner with you going forward.
And I'm just curious, with your experience so far, have you been able to, because with the therapy being produced and patients being treated, I would think that the company would already have a sense of, when you look at expression post-treatment, and how much do you need to see? I would think that would already be kind of being addressed in other diseases. So we're wanting to learn, even though it might not be relevant to our specific disease, we still would love to know if you've been able to determine, is it a tiny bit? Does it need to be a lot more? That kind of stuff.
Yeah, again, for hemophilia, it's very straightforward. If you have severe hemophilia, you have 1 or 2% of 100%, which is normal. And you get spontaneous bleeding in your joints and your muscles and your brain. 3 to 5%, you're mild, you have infrequent bleeds. And between 5 and 40%, you're mild, which means you don't have these spontaneous bleeds. So one of the first targets was, can we get you into the 5 to 40%? And in some cases we can. And then the real goal would be, can you get me 50 to 100? Because that is lab test-wise normal. And in hemophilia, the answer is yes. One sponsor has a phase 3 trial which gets you into the 50 to 100 range. So in hemophilia, it's a moving target. We can get you 5 to 40, and that's enough to live life, but you'll still need factor treatment if you're going to go surgery or have a trauma. And in some cases, in hemophilia A, they can actually get you into the 50, 100% range, which would be the goal. Why aren't we shooting for 100% for everybody? Well, the more of the gene therapy you give, the more you provoke the immune system. So it's not free just to give more and more gene therapy, because then you get a higher and higher immune response. So you have to find that sweet spot where you can get enough level to lead the life that you want to lead, but not so much that you provoke the immune system, and if the immune system attacks those cells which are making the protein, suddenly they're gone and the effect is lost. So each disease hopefully will have that sweet spot where we can get enough of the protein so the person can lead the life they want to lead, but not provoke the immune system to such an extent that it will come and attack and undo the good things we've done with the therapy.
I think that concept of the Goldilocks, sort of like being at the right level at the right time, I think is probably true across genetics. Mm-hmm. And I think that in addition to being personalized, hopefully what we'll end up with is more precision medicine as well, that we can understand, to your point, how much do we have to give. And we're still in the process of trying to find that out. And don't forget too, when you start altering expressions of genes, your body has natural feedback mechanisms that kick in to compensate, right? You know, that's the reason that we have homeostasis. That's the reason that we can function every day somewhat similar to the last day that we lived, because, you know, we stay within a buffer. But when we start altering, you know, downregulating a gene or upregulating a gene, you know, there are compensatory mechanisms that are designed to shift those balances, and those balances need to realign around a new equilibrium. And so we're all still trying to discover, you know, as we do these therapies, what's reasonable, and, you know, is it possible, first of all, and number two, is it reasonable? And then if it's possible and it's reasonable, does it do what we think it's going to do? And so, again, that's where I think registries and things like that really come in, because history will tell us more than anything. If you take a look at the patients who naturally have the hemophilia, you know, different grades, you can say patients who have 1 to 2% have these spontaneous bleeds, but above that, that's where we get those thresholds, is by looking at natural history and saying, this is actually what happens in real life, so it's reasonable to suggest if we could do it artificially, that should be a reasonable response.
Thank you. Okay, so this one, I think from one of our listeners, is probably a little bit of clarification rather than a question. My child has an excessively rare condition called generalized arterial calcification of infancy. She received gene therapy with residronate once weekly for 3 years. I have always been told the medication tricks the gene into behaving normally.
Pass on that. I haven't heard of that, and I don't want to say anything in an area I'm not.
I think it's more of the tricks the gene to behaving normally is maybe what. Does anybody in the audience have a? No? Okay. Apologize for that, listener. Next question. As a patient, if we do a clinical trial with gene therapy, we then can't use gene therapy in the future given a buildup of antibodies and other factors. How do we choose what and when to engage in gene therapy given we may only have one opportunity?
I am constantly humbled by the altruism of the community. Because that is, of course, the question. The ones that go first are always the pioneers. A very individual and personal choice.
So we talked about the immune system and the risks of neutralizing antibodies. Are there other risks that patients need to think about before deciding whether or not they should go ahead with gene therapy trial?
There are many risks. There are many risks. And I think those we have highlighted with the immune system are easily the ones that come to mind first. The other risk we also talked about is what if you do a therapy now and there's a better one in the future, but you can't take it because you've had here. The one thing I think everybody is most concerned about, and is why we need to follow patients for decades, is what is the long-term effect on the cell that now has this gene. The first history of gene therapy in the 70s and 80s had a problem where the gene was inserted into the chromosomes, and sometimes that insertion disrupted the chromosome and turned on a cancer gene. So the question would come, are we turning on any cancer genes when the gene therapy is given? And that's why many companies now choose not to do the insertion approach, but do the addition approach, so that these chromosomes are not interfered with by the gene therapy. Now, you can never be 100% sure in medicine or in life. So is it theoretically possible that we give this vector, which gives a gene that's supposed to be separate from the chromosomes, but somehow finds its way into the chromosomes? Theoretically true. A risk. So what do we put on the other side of the balance for that risk? Well, what we can say is the AAV platform we use is predominantly non-integrating. You can go into lab animals and take a look at their genes after they've been exposed to AAV and see if it's been inserted anywhere, and the rare places it goes do not seem to disrupt. And all of us may have been exposed. The older you are, the more likely it is. 50-60% of the population has been exposed and has had this AAV within them and is not associated with any known cancer of which we are aware. But that is really the reason that when patients take the gene therapy, it's unlike any other. If you're taking aspirin, you want to stop, you can. But if you're taking gene therapy, it's with you from that time on. So in addition to the immune response, one of the things we're concerned about is, are we putting patients at risk for a malignancy or a cancer? Previous experience has shown us if you stay away from integrating, those risks are low. But patients who take gene therapy really need to be in a registry and followed for the rest of their lives just to make sure that 10 or 15 years down the road we don't discover something. And as I said before, the patients are the pioneers. And I'm constantly humbled by their altruism to raise their hand and say, I want to do this, both for me and for the community.
So can I just make one other comment? Can I make one other comment? One of the other things that I think we're going to discover in the next 15 or 20 years will be, so I'm going to make up a disease, okay, which will unfortunately be lethal in 5 years. And let's assume we have a therapy for that. And we have somebody who's living with this disease now for 30 years. Look at HIV. HIV is a good example. Everybody died of HIV in the 80s. Now people are living with HIV for 40 years. And what we're discovering is that the HIV-infected individual doesn't have the same pathology as an otherwise healthy individual. There are certain things that chronic HIV infection does to that person over their lifetime that we never had to deal with before. We never had anybody survive 40 years with HIV before. But I think within the rare disease community, what we'll start discovering is there will be diseases that used to be lethal that aren't lethal anymore. And the next thing that will be asked is, well, then what then? And so I think, again, that goes back down not only for the risks of the therapies, but also to understand what does it mean to live with amyloid from the time when you're 30 and now you're 80. Nobody lives 50 years with amyloid. If you have early onset amyloidosis, you're dead. If you have it starting at 25, you're dead by 40. So what happens in 80? We don't know. And so that will become the next question, which will be, how does the disease progress in the absence of DTR in the liver, for example? We shut that off, now what? We don't know. So.
All right, it's about 12:30 now. I'm going to ask one last question on the live stream, and if anybody has questions to follow up, I'm sure Bartholomew and Alex would be happy to answer them after the session. Last question is, have any attempts been made by either of your companies to quantify the financial impact of a one-time gene therapy versus the current standard of chronic/palliative care for payers? If not, is this something that you think might support coverage?
Yeah, and that's actually one of Pfizer's initiatives. It's not just Pfizer, it's the whole pharmaceutical industry to try to quantitate what that means for patients. And we don't do that ourselves. We do that in partnership with patients because we have to be able to articulate what that means so that the financial community can place value and appropriately compensate for it. But that's something we and many other sponsors are doing now in concert with the patient community and patient advocacy, especially in hemophilia.
Well, thank you both for sharing your expertise and your passions. Very exciting stuff. And at this time, I'd like to invite everybody to just jump onto the mobile app and take a brief survey. And afterwards is the networking lunch that is hosted by Ionis. And there's a final break. If we could give, I know I was talking during that last round of applause, but one final round of applause for our guest. Thank you.